Good afternoon, hello, and welcome everyone to the 2020 PEGS Boston Virtual Rapid Novor Industry Presentation. My name is Anthony. I'm the Director of Business Development here at Rapid Novor, and today we're going to be talking about RePab, which is our polyclonal sequencing platform. We're going to be doing a walkthrough. For those of you that are unacquainted with Rapid Novor, we're going to talk a little bit about us, just to give you an idea of who we are, where we come from. Talk a little bit about RePab, the technology. Give you a quick primer. Don't worry, it's not too much work. Just a quick primer on mass spec-based protein sequencing, for those of you who've never seen it before. We're going to be talking about a proof of concept study that we ran to demonstrate the efficacy of our platform. And then we're going to be talking a little bit about applications in industry and some of the advantages of our platform, finishing off with a quick summary, right? That makes sense. Okay, so who are we? We're Rapid Novor, and for those of you that don't know, we've been in the game for a little over 5 years now. We have more experience than just those 5 years, I can tell you though. Certainly, our team is mostly comprised of bioinformatics and mass spec based talent that have a couple decades of industry experience. We've really focused in the past 5 years on specifically protein sequencing, whereas prior to that it was mostly like typical kind of core facility activities and proteomics as a software kind of space. But we've really in the past 5 years, like I said, focused on protein sequencing as a technology. And what has that yielded? Well, it's yielded many satisfied clients, luckily. That's why we're growing. We've got over 300 institutional users worldwide, including many of the top pharmaceutical companies. We're proud to say that we're the largest privately funded mass spec lab in Canada, and we've sequenced thousands of antibodies worldwide, which is, I'm going to bust out the laser pointer and say that this is our ReMab monoclonal antibody sequencing platform that some of you might be familiar with, where we are able to sequence any monoclonal antibody of interest as long as it's truly a monoclonal antibody, and you have permission to do so, where we can recover that antibody of interest, which has been really laying the groundwork for the subsequent service that we're discussing here today, RePab, which is for our polyclonal platform. The other service, the dedicated service that is new for those of you that are just kind of looking for an update from us, is NovaRig, which is a technology which is more geared towards quantifying the immune response and decoding exactly which antibodies are being produced in a sample of interest. Those of you, again, that don't know us that well, Wild, it was was a big break for us a couple years ago when we were able to actually differentiate between isoleucine and leucine on the basis of mass spectrometry, which was actually quite difficult prior to that, because you're using mass spec, which is obviously just using mass to differentiate between amino acids. Fun fact, isoleucine and leucine are the exact same mass, so being able to distinguish between those two amino acids using just mass spec was something to point out, and because we were the first team commercially to do that, and that was back a few years ago now. Okay. So this is the part you guys are a lot more interested in than me ranting about our services, I'm sure, is the RePab workflow. And so what we have been able to achieve thus far is use a rabbit in this context. And but the way that the projects run in general is like this. We have two arms that run in parallel. One starts with an antibody specific kind of fraction that goes for LC-MS platform. So we do blood, plasma, serum. We do an antigen affinity purification against that. Then we put that sample through our typical workflow of protein separation, digest, chemistry, and peptide chemistry. We're putting those samples through the LC-MS platform and generating peptide reads, and then leaving that in the library, where we combine the results from the second arm of our project, which is a PBMC extract going for NGS on the RNA, where we assemble the reads again into a transcriptome and create a library of reads that we acquired from the B cells, which is then matched with the LC-MS data, and which generates in the end a library of fully sequenced antibodies. Using our internal platform as well, we were able to achieve heavy and light chain pairing. This is not from the genomics. This is from our own techniques. And like I said, at the end of the day, what you're getting is sequences that were read directly from the combined proteogenomics data sets. So as we're going to get into the proof of concept, we're going to be showing a little bit of data. It's not that hard, but I just wanted to give you a quick primer on what to look for in our projects, but similarly for, I think, any proteomics sequencing kinds of projects. This is something you should be looking at. This is how you read our data anyway. So number one, the region that we're going to be referencing. In this case, you see the green, the orange, and the constant region. This is always how our reports read out. And what are those referencing? We have it annotated here. The green is for the variable framework, your orange is in your CDRs, and your constant region is in blue. Second is the sequence of interest. In this case, this is the full sequence for the heavy chain, variable portion of the heavy chain of the NIST map. And then below that is going to be the coverage. And so this coverage is for peptide reads that we got through the LCMS platform. And the different colors are referencing different enzymes that we use to cleave those antibodies at different points in order to generate the data. And what I'm showing you here with this alert is that, hey, this is the line for about 20x, which is to say 20 individual peptide reads for every individual amino acid. And that's important to note because that is when we have a very high level of statistical confidence in actually reporting on the antibodies that we're looking for. And so that's relevant in the next context, experimental evidence. But first, a little bit of intro, I guess, on what we did. So what did we do? We took a peptide antigen and we immunized New Zealand white rabbits. We gave them a boost. We extracted the B cells from the PBMCs and sequenced them on NGS in tandem with the purified IgG fraction that we got from rabbit sera, which we then sub-purified using a peptide-specific affinity column to try to get as pure of an antibody fraction as we could against the target antibodies that we're looking for. And so here are the results. In this case, we have coverage being shown for the first and third antibody that we generated because we felt we had good enough evidence to express 3 antibodies from this mixture. And I'll show you the results of that in a moment. But this is the coverage that we're talking about, right? And so this is in reference to the previous slide. 20x is something like here. And as you can see, we had really good coverage across these chains. For both the heavy chain 1 and the light chain 1, we had really quite good coverage across these antibodies, and we can get into more data if you're keen to talk about a project with us. But this is really what we're trying to demonstrate here: is that the level of coverage was quite good in these cases, and as a result, we felt strong enough to express them. And similarly, here, antibody 3, great coverage. We're happy to report on these cases and have them sent off for expression. And this is what those results look like. So as you can see, R1, R3, they bound quite well against that antigen. Whereas this is PD030 is the name of the original poly sera that we extracted. So as you can see, R1, R3 actually perform better than the original poly sera. And obviously our negative controls served us well here. They didn't bind, and that's good because that's what we wanted to see there. Except for R2, which was effectively not binding as well against the target. But nonetheless, what we were able to show was that R1, R3 were able to successfully bind against the target antigen with superior efficacy than the original poly mix, which is ultimately what we're trying to show here. And the reason that's important is because if you consider the case for this technology platform versus just a straight NGS approach, I think there's a couple places that we fit into that make us a little bit of differentiated by that system. So firstly, I mean, we're using NGS to sequence these antibodies directly, but in addition to that, we're also performing de novo sequencing against the monoclonals that we can see in the proteomics. And so for that reason, what we're getting out of that proteomics readout is functional antibodies, because we did affinity purify them, and then thus they bind against the target antigen, and whatever we're reading out of that situation is certainly going to yield functional antibodies. Beyond that, we're sequencing the most abundant antibodies. This is an inherent bias in our platform as well, is that not only are we doing like an antigen bias, but we're also doing effectively an expression bias, because the most abundant antibodies are going to be the easiest ones for us to sequence using our technology. So it's just another differentiator between us and an NGS readout, where you might get more reads for the number of cells, but not necessarily like the actual amount of protein being expressed in the organism of interest. We're sequencing native antibodies here, and that comes with an inherent advantage. I'm sure you guys can kind of fill in the blanks on your own here, but that was obviously an advantage of this platform versus like a traditional hybridoma or phage display. Not necessarily different than like an NGS platform is what I'm saying here. And I think the other key differentiator between our platform and an NGS platform would be the reduction in the number of variants to express, right? Like if you do a full B-cell sequencing screen, I'm sure you can appreciate you'll see a number of antibodies. At the end of that, in some cases hundreds, but are they all expressing antibodies? Are they all going to actually be functionally binding antibodies? And I think that's one question that we can answer using our platform, is really reduce the complexity of that kind of analysis by demonstrating that through proteomics. So we've had people come to us that have been quite keen to work on extracting therapeutic antibodies, either in autoimmune disease or cancer. I think have been kind of the more interesting ones that have received a positive response. An additional point of investigation is characterizing the immune response post-intervention, right? So certainly, in some cases, you know, in an intervention, whether that be a vaccine or an antibody treatment, there can be antibodies produced in response to that, and that's something that we can hopefully answer as well. Biomarker discovery and diagnostic development, and certainly converting a PAB to a MAB or MAB cocktail. There's a lot of polyclonal antibodies out there, a lot of tests that use them. I'm sure it would be great if we could convert those to standardized recombinant monoclonal antibodies or monoclonal antibody cocktails. And certainly that's an area of investigation that we're keen to explore. So in summary, you know, this is a novel method for determining the dominant amino acid sequence of the antibodies present in complex mixtures. At the end of it, we're going to provide a library of antibody sequences that are you know, useful in many applications, whether that be R&D therapeutics, diagnostics, or reagent antibody development. And you know, when it comes down to it, we're not seeking any royalties on these deals, so we're just looking to get some integration of our technology into the industry. So feel free to reach out to me. My name's Anthony. Again, been with Rapid from the beginning. So if you've seen us around, maybe you've seen my face, but if you haven't yet, please reach out and yeah, let me know if you have any questions. Looking forward to hearing from you. Bye now.